information about metabolism, well, that's pretty easy to come by. In fact, I make it really easy by providing a lot of graphics. But if you want to truly understand metabolism, appreciate its beauty, you need to actually engage with those charts, engage with the material. And so this is a spreadsheet that I use in order to kind of organize my thoughts when I was learning metabolism. Well, I mean, I still am learning metabolism because you never really stop learning because it is so complicated. But it's by engaging with this complication, and especially by engaging with how it's regulated, that you're able to then really learn metabolism, learn about the kind of logic behind how these pathways are arranged, how they're organized, how they're interconnected. And so this is just, I want to show you, walk you through this kind of chart that I have, this spreadsheet, and I'll share the link so that you can fill out this chart as you're learning metabolism. So basically all the answers, all the quote unquote answers are going to be in these charts or in charts in your books and things like this. And one of the key things about learning metabolism is learning how to read these charts, how to interpret the thing when it's showing you what like an allosteric inhibitor, something that's shown in like red, or an activator, something that's shown in green, being able to recognize examples of regulation, both feed forward and feedback, and to be able to integrate all of this knowledge together in a way that allows you to actively learn. And then the best part, or one of the great parts, is that then you're going to, once you fill this out, you have a great resource that you could turn to in the future. And if you want to test yourself, you can erase it and do it again. And so let's just take a look at some of these different tabs. So the one I'm on right now is the allosteric regulators. This is one of the key things that you want to focus on in metabolism, is the regulation. You'll see that a lot of the regulation, a lot of pathways are actually going to be regulated by the same allosteric regulators. So remember, allosteric regulators are like small molecules that bind to an enzyme and change its activity. It can activate it, or it can inhibit it. And so maybe you even want to put this activate in green and inhibit in red to make it even more obvious. Often you're going to have the same molecule activating some pathways and inactivating the reverse pathways. But you'll also see that some pathways, some steps of pathways are activated by the same molecules and some are activated by different molecules. And by focusing on the similarities and differences, you can learn key things about metabolism, such as the logic of the regulation and what changes in metabolism you might expect under various cellular conditions. If we take a look at some of the molecules that you see as allosteric regulators in metabolism, they tell you about different things. For example, ATP, AMP, and ADP, they tell you about the energy status of the cell. If you've got a lot of ATP, well, that's going to indicate high cellular energy. And if you have a lot of AMP, that'd be low cellular energy. Similarly, you would have your NAD and your NADP and your FAD, and all that stuff would tell you about kind of the oxidation status of the cells. Whereas these kind of intermediates from the various pathways we see, those tell you about the activity in those pathways, maybe what's building up, what's in short supply. And so all of these kind of make sense and tell you different things. So when you're learning these different pathways, take notes on what is going to be regulated by what. So you can put down what the enzyme is and what the pathway is involved in. For example, I filled out a couple of rows. If we look at ATP, well, as I mentioned, this is going to indicate high cellular energy. In this other note column, I just put that it's produced by electron transport chain, so that would be our aerobic production of it, as well as substrate level phosphorylation. So when we're just making ATP or we're making GTP, but right now we're making ATP, but you can convert GTP to ATP. Bottom line is you're making one of these NTPs without using the electron transport chain. So like the ATP that you're making directly in glycolysis or in the citric acid cycle, not requiring the actual going through the electron transport chain, but simply taking a phosphate and sticking on a dinucleotide. Okay, so the ATP, we got a lot of energy. What do we want to do? Well, we don't want to break down molecules to get more energy. Instead, we want to use that energy to build things up. So do things that are anabolic. We want to, in this case, activate gluconeogenesis by activating fructose 1,6-bisphosphatase, or FBPase1. On the other hand, well, what do we want to inhibit? We said we didn't want to break down stuff, so we want to stop glycolysis, which we can do through PFK1. We want to, and we can also do through pyruvate kinase. We want to stop going into the TCA, so we can inactivate pyruvate dehydrogenase, and we want to inactivate the DCA itself, so we can inactivate isocitrate dehydrogenase. And well, when those energy levels get low again, well, here our AMP is going to act as an allosteric modulator, and it's going to basically do the opposite. So it's going to activate glycolysis by activating PFK1, activate entry into the TCA by activating our PDH, activate lots and lots of different catabolic enzymes as well at the transcriptional level, so to make more of them, and do the converse for your anabolic enzymes or their transcription. How is it going to do all this? Well, it turns out that we have AMP-dependent kinase, this AMP-dependent kinase, AMPK, and it's going to be activated by AMP. And so if you have a lot of AMP, this is going to allow this one molecule to sense that and then go and activate a bunch of other stuff and inactivate a bunch of other stuff. And that AMP is going to inactivate that fructose 1,6-bisphosphatase, or FBPase1, that we had activated by our ATP. So you can see that we have inverse regulation of pathways depending on the energy status. So if you have high energy, you want to inhibit the breakdown for more energy and activate the piecing together those molecules, building them together. And if you have low energy, well, now you want to activate the things that'll give you more energy and inactivate the things that take energy. And so that's how you can do this. And then I just put as a note that AMP is a more sensitive indicator of our energy levels than ADP. And that goes back to that whole thing about how the AMP levels are going to be lower than the ATP levels. So small changes are like small numeric changes in them will have a larger percentage-wise difference, as well as the fact you can kind of interconvert between the nucleotides and then have the AMP be more sensitive than the ADP. But anyway, that's getting a little off topic. Just that's the sort of thing that you can put in your other notes column. This was allosteric regulation, but we also see a lot of hormonal regulation. It's important to note this too. This is probably one of the harder tabs to kind of just figure out how you want to organize it. So I gave my students some hints, as well as some examples of how I would organize things. But this is your spreadsheet. Make it how you want it. Do however it helps you learn. But kind of focus on, these are kind of the main hormones that you'll see, but there could also be others. So insulin, glucagon, adrenaline, or epinephrine, same thing. Cortisol, what type of hormone are they? Where are they made? How is their making regulated? What tissues are affected? And then within those tissues, so for each thing, you choose a target enzyme or other protein, and then how is it affected by this hormone? And in what tissues is that happening? And then how does that come about? So if we say, okay, well, our insulin, it's a peptide hormone, it's produced by our pancreatic beta cells. You make more of it when you have high glucose and ATP concentrations, and less of it when you have glucagon. One of the things that it does is it increases glucose uptake by increasing the transport of GLUT4 to the surface of the cells. And where does that happen? Well, it happens in the cells that have the GLUT4, and so that would be your muscle, heart, and adipose tissue. And well, how does that come about? Well, you have the activation of AKT, so that's the same as PKB, remember, through that PI3K pathway, followed by complicated stuff involving G proteins, RAC1 and RAB, which I don't make my students know about. So again, because the hormone one was a little more complicated, I gave my students more of the information, but this is only partway filled out. So you fill out the rest, and this will help you learn. And then kind of just like a summary table, bottom line, end of the day, what effects does insulin and glucagon and adrenaline have on the various tissues? And then you can think about, well, why? And why would it have one effect in one tissue and another effect in another tissue? Thinking about the key roles of those tissues. And so that's important to focus on, is kind of what's the function of these different tissues, because they all have different functions and some of them have different weaknesses. So like your liver, it can do glucogenesis, but other cells can't. It's got that glucokinase rather than hexokinase. And so it can sense and respond to blood sugar levels. It can process and distribute fuel to the body in many forms. And it prefers to use fatty acids, which kind of makes sense that it doesn't prefer to use glucose, or else it would just we would just eat up all the glucose that when instead of freeing it for the other cells. And so then you can have like other notes, kryptonite, what it can't do, what it can do. So remember that, say your erythrocytes, so your red blood cells, they don't have mitochondria. So they can't do the citric acid cycle, and things like that. Then for each of the key kind of pathways, what are the regulatory points of those pathways? When do you want those pathways to be active? And like when might you want them to be inactive? And so when we talk about how do we get it to be active, well, that goes back to thinking about our allosteric regulators as well as our hormones. So as you learn about different pathways, the key things to focus on are these types of main things. So like, what are the key regulatory points at which, say, we were having those enzymes be regulated by our covalent regulation, such as through hormone triggered phosphorylation or through our allosteric modulators? Think about when you want that to be active, how you're going to go about activating it, where it's going to occur, both in the cell as well as in the tissues. Because some of the things are only going to happen in certain tissues, and that's more important that you kind of like know which are ones that happen only in certain tissues than trying to know like whether it happens in this type of cell or this type of cell when you have like a bazillion types of cells in your body. For that sort of thing, just like Google it, but know about those certain pathways, such as your urea cycle, such as your gluconeogenesis, such as your glycogenesis and glycogenolysis, that only happen in like certain cell types. It's like your lipogenesis and stuff like that. For each of these specific pathways, then, what I have is I organize things like this. Again, if something makes more sense to you to do it a different way, then do that. But these are the things that I think are most important to focus on when you're learning about a pathway. And so this is just like a template, but here, for example, is a carbohydrate metabolism tab. I've gone ahead and for the carbohydrate metabolism, because this was like the first one that I did with my students, I gave them I gave them some of this information, and then I'm having them fill out the rest. But for the different pathways, you can see that a lot of these pathways, these enzymes, so basically there's one row per enzyme. I probably should have put the enzyme name and abbreviation row first, but it makes more sense to me in my mind this way for some reason. But if it makes more sense to you to basically swap these, then that's what you can do. And that's totally cool. But basically, you have for each row, there's a different enzyme. And then it's what pathway or pathways is it involved with? Because you can see, well, some of these are involved with glycolysis and gluconeogenesis, whereas some are only in one direction. And then you can see that some of them are going to be involved with various different pathways. And then for each of these, the enzyme, what's the reaction it catalyzes? So write it in one direction, but you can see that, well, if the forward direction, it would be involved in glycolysis, and then the gluconeogenesis is involved in the reverse direction. This is again with one of them that's only like partway filled out. And so, but you want to go through and see, okay, well, did I use NADH or did I use NADPH or FADH2 or did I generate them? And the same thing with ATP. Is it reversible or is it irreversible? So you can see that the ones that are kind of reversible, these are involved in multiple, in like the forward and reverse directions. They might be involved in different pathways, like glycogenesis and gluconeogenesis. Whereas others, like are only going to be involved in kind of like a single direction, the ones that are irreversible. What are they regulated by positively and negatively? And this goes back to our idea of our allosteric regulators. And so, but in this spot, you're seeing how not only does it regulate that one enzyme, but it's also going to regulate these other enzymes. What do those have in common? What pathway? Are they in the same pathway? Are they in different pathways? Are they pathways that you both want active at the same time? Are they pathways that you want inactive at the same time? These are the sort of things that you want to focus on. Where are they happening in the cell and in the tissues? and kind of like what is there, like any other notes that you have. So when sometimes in metabolism, you're just thinking and thinking and you're stuck and then aha, things click into place. And so if you have kind of those aha moments where you figure out things that help make things click for you, then you can kind of like stick it in your other notes category. So this is a carbohydrate metabolism tab. So I covered like glycolysis and gluconeogenesis, glycogenolysis and glycogenesis, as well as the pentose phosphate pathway. But then there's also, we talked a lot about electron shuttles and the citric acid cycle. That's what we're about to cover. I'm really excited for that. Then we'll get into lipid metabolism. Because that one's a little more like complicated because there's like multiple enzymes and stuff. I just put some of the key ones that I want my students to focus on, kind of the major ones and the major regulated steps. And then we'll talk about amino acid metabolism. We'll talk a little bit about some other metabolism. And so this is kind of like the doesn't fit anywhere else tab. So things like our ethanol metabolism. And then kind of looking at how are they all interconnected. So we'll see, you see when you're learning about metabolism that you have a lot of points at which there's multiple directions that you can go. So if you think back to your charts and diagrams, there's a lot of these places where you kind of have more than just two arrows linking off of something. Like look at pyruvate, like it's got all these arrows hanging off of it, as well as your acetyl-CoA, your glucose 6-phosphate, your glyceraldehyde 3-phosphate. These are kind of like key points at which you can go lots of different directions. And so you want to think about, okay, well, if I have this molecule, what direction might I want to go? What enzyme would I use to get there? And where would that be happening? So kind of for these are some of the intermediates that I have in my chart, but there are other intermediates to include. And so these are just some of the examples that I give. For each of them, kind of talk about, well, how many carbons does it have? What enzyme can do what to it? What's the reaction catalyzed by that enzyme? What pathway or process does that happen in? And what's kind of like the role or the function? So for example, if you have dihydroxyacetone phosphate, your DHAP, you could use aldolase to piece it together with with a glyceraldehyde 3-phosphate in order to give you F16BP, or you could basically isomerize it to glyceraldehyde 3-phosphate, basically to do the glycolysis direction, or to go from glycerol to lipids to interglycolysis for further breakdown. So you could also use glycerol 3-phosphate to basically take that DHAP and reduce it to glycerol 3-phosphate, which is going to be involved in the glycerol 3-phosphate shuttle as well as triacylglycerol synthesis, because now you can use that glycerol 3-phosphate to make the backbones for your triacylglycerides, your phospholipids, and so that would be for your lipid synthesis, and for your for the shuttles, that would be getting reducing equivalents from the cytoplasm to Q. And again, these are all kind of things that you can find in these charts and these diagrams. So when you look at your shuttles, you can see that we're talking about things happening over here. So those are kind of the sorts of things that all of this information is here. And all of this information, if you just look at it, it can look really complex and really confusing. And if you could look up answers in here, but learning metabolism isn't about looking up answers, and it's not about memorizing anything. It's about understanding things. And so my hope is that by providing these resources, by providing these charts and this information, you waste less time Googling things, Googling these charts, Googling what enzyme does what where. And instead, you kind of have a central location for this information, and then you will gain the knowledge of the pathways by really interacting with them in an active and engaging way, and taking notes, making things that are going to be helpful for you down the line. But so focusing on kind of the engagement with the material rather than just finding the material. So I hope that this is helpful for you, and I will share a link, and happy learning.